Hey, in this video we're going to be getting the reverse complement of a DNA string. Luckily, computing this is rather trivial. Let's begin with a sample DNA sequence. As always, it's composed of 4 nucleotides. Next, let's define a reverse complement function that accepts a DNA sequence as an argument. We'll then define a hard-coded mapping of each nucleotide's complement. This can be achieved by using Python's built-in dictionary data structure. After, we'll create a list of complements by iterating over each nucleotide and getting their respective mapping from the dictionary. We can then join this list back into a string by using Python's built-in join method. Lastly, we'll reverse the string. Instead of using an unnecessary variable, we'll simply return this result directly on the same line. Now that the function is complete, we'll call it by passing in the sample DNA sequence and printing the result to the console. If we take the reverse complement twice, we'll notice that the string maps to the original sequence. Hope you enjoyed the video. Thanks for watching.